So today I'm pleased to present Ludwig Geislinger, who is a postdoc in my lab and a true expert at gene set enrichment analysis. It's something he's been working on extensively since his time as a PhD student. He has published, I think, the most user-friendly enrichment analysis package in Bioconductor called Enrichment Browser. And you'll be hearing about that today, and is also in the process of doing extensive benchmarking of all of these different enrichment methods to help clear up somewhat more of the differences between them. So with that, I think I will let Ludwig get started.
All right. Thank you for this introduction, Levi. And with this, hello and welcome. Good afternoon. I hope we fix these audio problems. So today's workshop is about gene set analysis. My name is Ludwig, as introduced, and what we're going to do in the next 2 hours, I want to give a bit of an overview about like existing methods for enrichment analysis, not only for gene sets, but also like pathways and regulatory networks, as well as in the end also like genomic regions. In the context of gene expression data as obtained with microarrays and RNA-seq. So I posted quite some introductions here on the meetup page for requirements. If you want to follow along that workshop, please make sure that you're using the corresponding R version and Bioconductor version. There can be all kinds of issues when not using the right version. So please make sure that you have the current R release 3.4.4 and the current Bioconductor release version 3.6 installed, and make sure that you have these 6 packages. These are Bioconductor packages, the enrichment browser, the regioner, 2 annotation packages, and 2 data packages, which you can install in the typical way via BioC Lite. And if you actually want to see the material for this workshop, this is available here, where you have on the one hand side the RMD source file, and on the other hand also like a fully rendered HTML that you can just open in a browser. Everybody fine with these requirements? Well, any more time needed for setup? I think it looks good, so I just assume at this point that you have all things set up, and we can start looking at this workflow here. So this diagram shows like what is, I think, the standard workflow for gene expression data analysis, where you carry out a transcriptomic study, a high throughput study, either with microarrays and RNA-seq, then carry out some pre-processing in order to get rid of technical effects and have the biological signal that you then want to analyze either gene by gene. So for differential expression, for example, between 2 sample groups in a clinical setting, between, for example, diseased patients and healthy patients. And the main focus for today, I mean, we could speak about these 3 steps here, each of them like for a whole workshop, but today we want to talk especially about gene set analysis, as well as network analysis. So, and what we will encounter over and over again, that we define these gene sets according to the gene ontology or the CAG pathway database. So what I'm assuming here that you're mostly familiar with like high throughput transcriptomic studies, you know a bit of normalization and also about differential expression analysis. I think we also had a workshop already here in the context of the Bioconductor meeting. So why do we do that? Well, if we find certain gene sets which are representing molecular functions or pathways enriched, it typically helps us a lot when it comes to biological interpretation of these high throughput experiments. And in the best case, we might even be able to draw some conclusions on the phenotype that we are investigating here with these experiments, which could be a disease. So where does this whole enrichment thing comes from? Well, as I said, we want to test whether there are like known biological functions or processes, for example, as defined via Go or CAG, are overrepresented, aka enriched, in an experimentally derived gene list. So which could be a list of differentially expressed genes, okay? And a standard reference in this context, if you want to get a little bit of an introduction into the statistical background, is this nice paper from 2007, which I highly recommend. So let's consider a little example here. We have a transcriptomic study where we measured gene expression either via microarrays or RNA-seq. And we have like almost 13,000 genes measured in total. Okay, and we have tested all of them for differential expression between two sample groups. Let's say diseased and healthy. And 529 out of these almost 13,000 genes were found differentially expressed. Okay. Further, among these 530 differentially expressed genes, we have 28 genes which are annotated to a specific functional gene set that we are investigating, which in total contains 170 genes. So statistically, this corresponds to a 2x2 contingency table, which we can construct here in R with the matrix command. Rendered, it looks like this. So we have 28 genes out of the 170 genes which are differentially expressed. And in total, there were like 529. And this overlap between the genes and the gene set and the differentially expressed genes, we can test or assess for statistical significance based on the hypergeometric distribution. Okay, so our test statistic is the overlap between differentially expressed genes and our genes in the functional gene set. And in statistical terms, this corresponds to a one-sided Fisher's exact test. So if we're applying a Fisher's test here, asking how likely is it to obtain an overlap as large or larger, it would give me in this context like a significant p-value, significant enrichment. So very easy, intuitive, basic principle, but still like the core of what we are talking about when we're talking about enrichment over this talk and in general. And you can also keep in mind that this principle is also underlying the existing major public and commercial enrichment tools such as David and Pathway Studio, which I guess you have already heard about. So for the largest part of this workshop, we are talking about transcriptomic data, and gene set enrichment methods have been mostly developed and applied on transcriptomic data, but these concepts are like currently extended and applied also in other fields of genomic and biomedical research. And I just added here some references for you in the context of other omic data. So for example, enrichment analysis in the context of proteomic or metabolomic data. And later in this workshop, we also talk about enrichment of genomic regions. Okay, but let's start with enrichment analysis in the context of gene expression data. And this will be, as Levi nicely introduced, on this particular package, the enrichment browser package, which tries to implement this little workflow here. So it gives you some standard usage, some well-established bioconductor packages in order to set up your data over the pre-processing and the differential expression step and have it ready for the various enrichment methods, either with respect to gene sets or gene networks. So how does this pipeline look like? Which bioconductor packages are involved here? So the typical two inputs for gene set enrichment analysis here on the left-hand side, expression data as obtained from microarrays and RNA-seq. There is functionality for normalization for microarrays based on the lemma package for RNA-seq data. based on the excellent RNA-seq package from David Riso. Then there comes the differential expression analysis, which is for microarray data, again, based on the lemma package. For RNA-seq, we also have two other alternatives, which are very popular, the EDGE-R package and the DE-seq package. In the end, we end up with a table of all genes tested for differential expression between the sample groups in our experiment. And the second input for gene set enrichment analysis are gene sets, which are either defined on Go or defined according to KEGG. And these gene sets can then tested for enrichment in the differentially expressed genes using different set-based enrichment analysis methods. And we will mainly talk about these two, overrepresentation analysis and gene set enrichment analysis today. A third input is required if you also want to do like network analysis, so you want to take into account also interactions between the genes in your gene sets. So you're not assuming them as independent, but rather you take specifically into account whether genes are activating or inhibiting each other. Then you can also, like as a first workaround, compile a network based on interactions which are there in the pathway diagrams from KEGG, or in case it is something there, so for specific organisms, you also might have transcriptional regulatory interactions annotated and collected in specific databases. So for example, for E. coli here in the Regulon DB, or for yeast here in the yeast tract. And then you have a network, and then you can take the three inputs, the expression data, the gene sets, and the network for some network-based enrichment analysis. And we will talk mainly about gene graph enrichment analysis and signaling pathway impact analysis today. No matter which method you apply, whether you apply based on the set-based paradigm or on the network-based paradigm, you end up with a ranking. A ranking of these Go gene sets or these KEGG gene sets, which tell you, okay, which of these gene sets are enriched in my data according to my statistic that I define in my method. And I can then browse, explore, and visualize these gene set rankings, especially the enriched gene sets, individually for each method. And here in this package also, you can combine like the rankings between methods. And in the end, you get an HTML report that we are going to explore a little bit more later on. So let's start with loading the package and then go a little bit into analysis with the enrichment browser, but just like beforehand, in order to make sure that we're all on the same page, a little bit of vocabulary, but I make it very brief because I think many concepts are very well established. And I think you also can, if not, read through this text, which I have provided you here and in the RMD file on the GitHub repository. So what are gene sets? Gene sets are just simple lists of functionally related genes, but I do not further specify here the relationships. So if I take a look at a little example, increase the font here a bit. So somebody back there, can you see that, what I'm typing here? Yeah, great, perfect. So I already have the example here. A gene set can be as easy as carrying out this arc command. So we have here a gene set of 10 genes, okay? And I do not specify any interaction between these genes, whether they might be phosphorylating, activating, inhibiting each other. I just say, okay, this is a gene set, and these genes might have something to do with each other or not. They might be on the same chromosome, they might be in the same pathway whatsoever. So what are pathways? Pathways can be like interpreted as specific gene sets, and there are typically two different kinds of pathways. One are like metabolic pathways. So if we take an example, let's take glycolysis, looking it up in the KEGG database. And selecting here for human, I increase the font again. So these metabolic pathways define like metabolite conversions, yeah? In glycolysis, it's about conversion of glucose in the end to like a pyruvate. And as you can see, there are like different enzymes which are catalyzing these conversions. And these enzymes are encoded by genes. So if we would think about this as a gene set, we could collect all the genes which encode the enzymes here active in this pathway. What is also especially interesting in the context of network-based enrichment analysis are signaling pathways, which differ from metabolic pathways in the sense that they indeed define interactions between gene products. Okay, so as you can see here, all these green boxes, these are like proteins, so gene products, and you have all kind of interactions here. Yeah, some phosphorylation. So you have some signals which are recepted at the cell membrane, which causes an activation of a cascade, and in the end, some activation of gene expression. So some activation of transcription factors, which would interact with the DNA and cause gene expression to happen or not. And of course, these interactions are very interesting, and these are also like assessed and evaluated in these network-based methods. So going back here to these definitions, so gene regulatory networks are, so pathways, signaling pathways are specific cases of gene regulatory networks, but little gene regulatory networks. Gene regulatory networks, in my understanding, is a more broader term for like collecting all a gene regulation on a larger scale for an organism. Resources that we are talking frequently about is Go and KEGG for gene sets. I don't know whether I indeed have to introduce that. Who has never heard about the gene ontology? Okay, great. Who has never heard about the KEGG database and wants me to say some introductory words? Okay, so does that only hold for KEGG or also for Go? Just KEGG? Okay, so in comparison to Go, where we indeed have like these ontologies, so molecular function and biological process and cellular component, in KEGG we have these pathway maps, which I just showed. So these are specific little processes in which we have either like these metabolic pathways or these signaling pathways where we have interactions between the genes. And the good thing is both of these resources are manually curated and long-standing. So they both were established like around 1999. So almost 20 years of curation. So they represent some very nice resources for genomic analysis. Okay, the two methods that we want to talk about for doing enrichment of like Go and KEGG gene sets are, on the one hand, this overrepresentation analysis, which has the principle which I introduced in the very beginning. So this is based on these Fisher's exact tests. So we are testing whether a gene set, a functional gene set, a Go term, or a KEGG pathway contains disproportional many genes of significant expression genes. So disproportional many differentially expressed genes. As is already given in this definition, the overrepresentation analysis concentrates on the differentially expressed genes. And of course, they are only like a small subset of all genes which are measured via these transcriptomic assays. So that's the good thing about gene set enrichment analysis. You do not restrict your analysis only to differentially expressed genes, but you really work over the whole a set of measured genes and you test whether genes of a gene set rather accumulate at the top or at the bottom of the full vector of genes that you have measured. Yeah, so you sort your genes by a statistic, for example, fold change, where you have like at the top the most positive upregulated genes and on the bottom the most downregulated genes. And then you have your gene set, which is like a sample from this whole vector. And you ask, is this a uniform sample from this vector, or is it rather enriched or at the top or at the bottom of this vector? Okay. Okay, so there's a little bit of confusion with terms in the field of gene set analysis, also like gene set enrichment analysis. There is this particular method here from 2005, which we are using over and over again. However, like the term gene set enrichment analysis is also like a very general term, which rather is like an umbrella term for all of these methods which do these kind of concepts. So I hope this is always clear from the context, but I try to make it super sure and super clear. Some authors say instead of gene set enrichment analysis, they say gene set analysis in order to like distinguish between the general strategy and this particular method. However, to make it even more complicated, there is also a specific method named gene set analysis. So this distinction is not as easy. Okay. An important point when it comes to gene set analysis is which null hypothesis are you actually testing from a statistical point of view. And in this paper I have referenced in the very beginning, it points out there are like two main null hypotheses which are tested here, and it's always good to know when applying one of these methods which null they are actually testing. What are you actually testing here? So the one thing is this competitive null hypothesis, which is the genes in the gene set which I'm currently analyzing are at most as often differentially expressed as the genes not in the set, okay? So this is exactly what I have outlined with this 2x2 contingency table. We are testing the genes in our set for overlap with the differentially expressed genes as compared to the background. How often does the background overlap? Okay, so this is this overrepresentation analysis as a competitive null hypothesis. More stringent is like the self-contained null hypothesis, which says, okay, no genes in my set, which I'm currently analyzing, are differentially expressed. So a single gene is already enough to turn this set significant. Yeah, so you have to ask yourself, what are you interested in? Sometimes it might be indeed relevant if you want to see, is there anything going on in my gene set? And then the self-contained might be very appropriate. Most often you're rather, you have a certain fraction of differential expression in your set, and you're rather interested, is there more than you would have in the background? Okay, I already pointed out that there are some issues with these overrepresentation analysis, where the most prominent issue is, okay, you need to distinguish between differentially expressed genes and not differentially expressed genes. And how do you do that? Well, you do a test, and then you say, okay, for example, my p-value below a certain threshold, these genes I consider as differentially expressed. Okay, but this is of course a rather arbitrary classification, and the good thing about gene set enrichment analysis that we do not need to do this. I do not want to overstress these aspects here. I think overrepresentation analysis definitely has great applications, and you also need to take keep in mind that it's a very simple concept which is very generally applicable. Yeah, so it can be also applied in all kind of contexts where you just have gene lists. For gene set enrichment analysis, which have some superior aspects statistically, you, however, need the complete expression data matrix. Okay, and this is especially for like experiments not based on transcriptomic data, not there. So gene set enrichment analysis cannot apply it in this context. So beside the statistical definition, there are very intuitive definitions according to this reference here, which just do it like according to the timeline by how these methods were developed, and it divides it into 3 generations. So the first generation are these overrepresentation methods, variation of it based on the 2x2 contingency table. The second generations are methods like gene set enrichment analysis, where you have a local statistic for each gene in the gene set, which could be a fold change or a T statistic or something like this. And you kind of like accumulate or combine this local statistic for each gene to a global statistic for your gene set. And you call these functional class scoring. So the functional class, a gene set, is scored. And then the third generation are these topology-based methods, also network-based methods, which like explicitly take into account interactions between the genes as defined, for example, in such KEGG signaling pathways or gene regulatory networks. That sounds very appealing. To take into account these interactions sounds like realistic. You know, these genes interact with each other. However, they also come with some problems. So for example, some features might not be detectable alone in transcriptomic data. If you think, for example, about protein-protein interaction, a phosphorylation between 2 genes, you do not necessarily see in transcriptomic data, okay? And well, network knowledge is also not as nicely curated and stored as, for example, gene sets are. So we have standard databases across many, many organisms for gene sets like in Go and KEGG, but we do not have such standard repertoires or standard databases for networks now. And that makes network analysis a little bit difficult from time to time, a lot depending on the organism under investigation. Okay, and this is a little side note. So there are all these different methods, and many of them have great advantages. Many come also with some limitations. So it's a good idea to not only apply a single method, but rather like inspect across methods your results and make sure that you really like accumulating confidence in your results there. Okay, so far to the introduction. Any questions here at this point? Okay, so I would say then it's high time to get a little bit to the practical aspect and to really make use of the enrichment browser package. And for that, we are looking at 2 different data packages. A data package for microarray data on leukemia and a data package for RNA-seq data on asthma. So for the microarray data set, we take here the ALL package, which is available in Bioconductor, and it's investigating patients with acute lymphoblastic leukemia and a frequent chromosomal defect found among these patients is a translocation in which like parts of chromosome 9 and chromosome 22 like swap places. And this results in this cancer driver gene, a fusion gene, the BCR-ABL gene, which is exactly created by this translocation. So it's positioning the ABL1 gene on chromosome 9 to a part of the BCR gene on chromosome 22. So how to load the data? I think this is very straightforward. So you load the L package and then the data set, looking at the contents of this data set. So you see it's an expression set. So that's the traditional way of how microarray data has been presented in Bioconductor. It is more and more replaced by summarized experiment, which is in many respects a very similar structure. And we're going to use this later on as well. So what is the expression set? It's basically an assembly of 3 matrices, one matrix which stores the expression data, one matrix which stores information on the patients, on the samples, and one matrix which stores information on the features, so typically genes. So what do we do with this data? So first of all, we select a subset of patients in this data. We select here only leukemia patients with a B-cell leukemia, and which either do have or do not have this fusion gene, as it has been previously described in this Bioconductor book. So how do we do this? We do a little bit of grabbing here on the phenotype variables, and also ask, okay, do they have the fusion gene BCR, ABL, or are they cytogenetically normal NEG? And then I say, okay, from this expression set here, I only take these samples which fulfill these both conditions. Okay, so it's here are the columns, which are the samples, and before the comma are the rows, which are the genes. So I basically say, give me all the genes, but only take this subset of samples. So what we have, if I ask about the dimensions of this expression set now, so we have 79 patients which fulfill these conditions, and we are investigating here almost 13,000 probes on this microarray. So it's a particular Affymetrix platform. So let's look a little bit of these expression values. I said the expression set has 3 matrices. One of them is the expression matrix, which can be accessed via expression here. And I just look at the first 4 rows and first 4 columns of this expression matrix. And I see here in the rows, the probe identifier of this microarray, and here are these 4 samples with these rather uninformative identifiers. And here I have the expression level of this particular probe for this particular patient, and these are like intensity measurements obtained from microarrays. So what are these probes about? Well, I'm not diving too much into that here. It's just like, okay, these probes are parts of a gene which are like hybridized onto the microarray, and somehow we have often more than one probe for a gene. So what I often want to do is I want to summarize the intensity levels from several probes which are annotated to the same gene. And I can do this using this one particular function from the enrichment browser, which says, okay, summarize all this probe level expression on gene level, because I want to do differential expression analysis and gene set enrichment analysis. So I want to talk about expression levels for genes. So, and if I do this, these row names change. They are not probe set identifiers anymore. They are rather Entrez gene identifiers now. Yeah, so these names correspond to Entrez and CBI gene identifiers, which are all digits. So if I look into this now, I see here, okay, these are now genes, and I have these summarized expression values in here. Yeah? A very good question. So this is pointed out exactly here, though this is a bioconductor annotation package, which is there for most or frequently used platforms, which defines these mapping. And the good thing is this function automatically detects which platform this is by looking into the annotation slot of the expression set. So, so the expression set has a slot which is called annotation, which gives the platform, and the function checks whether there is a package for this platform. If there is such a package, it loads this package from bioconductor and carries out the mapping from probes to genes and the summarization. Does that answer the question? Okay. Further questions?
Is the ALL data set in a package called ALL?
Yes. So this is the data set. Oh, yes, please.
Yeah, I mean, mine is not finding this function of Protein.
I'm sorry?
It's not finding this function Protein set, even though I know that this HT295 or all other packages.
Yeah, which R version and bioconductor version?
I run it on RU0, both versions as you suggested.
R 3.4.4 and bioconductor 3.6. Levi, do you want me to wait on this point, or you figured this? Thank you so much. And I continue in the meanwhile. Great, thank you. So this is the L data set. This is the microarray data set. And I also said, and I guess most of you are here to learn about RNA-seq data, which is more and more replacing microarray data. So we also consider at the same time another data set. I just want to point out, why do we talk about microarray data then if it's not that relevant anymore? Well, it is very relevant because many of these gene set enrichment methods have been developed for microarray data, okay? And microarray data and RNA-seq data are, show different aspects when it comes to like distribution of the data. So all these gene set enrichment methods have some statistics incorporated which assume a normal distribution of the data, which does not per se holds for RNA-seq data. So there are several adaptations necessary also in gene set enrichment analysis when you're dealing with RNA-seq data rather than microarray data. So let's take a look at this other data set. So what is that? These are like transcriptome profiles for 4 primary human airway smooth muscle cells and 2 conditions, a control, and treatment with a certain drug, okay? And we can go through the exact same steps as we did for the leukemia data set. We load the package, we load the data set in that package, and then we do a little bit of pre-processing here. So what do I do here? I select from this data set, which is a summarized experiment, so a very similar but more recent structure, overworked for support for genomic regions and also like efficient dealing with big data, but all these slots are very similar to an expression set. So I also can access and subset a summarized experiment in the rows and in the columns, so genes or samples. And here I say, please keep only the genes which have an ensemble gene identifier. Okay, all others, please remove. And this ends up like in 60,000 genes for which there is an ensemble gene identifier in the data set. And as I said, we have 8 samples, 4 untreated and 4 treated with a drug. Okay. Mm-hmm.
Enrichment browser and the region are libraries at this point?
they need the regional, the enrichment browser library at this point. So for a summarized experiment, the expression matrix is accessed via assay. So beforehand, for an expression set, we had EXPRS. Here, for a summarized experiment, we assess the expression matrix via assay. Again, we are seeing here the genes in the rows and the samples and the columns. And now we do not have intensity measurements as for microarray, but rather read counts. So we have how many reads have been mapped onto this gene for this sample. Okay. So far to loading the data and pre-processing the data, how about a differential expression analysis? I skip here a bit over normalization. This is a very important topic where I think also a lot can be said about, but we really want to talk about gene set enrichment analysis here. So I rather start at the point we have normalization carried out and we can compare between samples. There is, however, functionality, easy convenience functions for normalization as shown beforehand in this workflow, also integrated in the enrichment browser that you can easily apply. So, thank you so much. Great. What was it?
A combination of things.
Okay, good, good. So
yeah, speak up if you have problems you've got.
Yeah, absolutely. Okay, so differential expression analysis can be done based on various packages. As I said, the lemma package is a standard package that you want to use here, either for microarray data or for RNA-seq data. In case of RNA-seq data, you would apply the boom transformation beforehand, which kind of assures that your data follow some distribution aspects as you would observe for microarray data, and you can then apply a lemma t-tests. On the other hand, there are like also packages like Azure and DE-seq, which really work on the read counts, which rather follow a negative binomial distribution. And I guess in the differential expression workshop, we have talked a lot about this, right? Yeah. Okay. So for carrying out the differential expression analysis, there are some basic things that you have to annotate to your summarized experiment under investigation. And especially there's one important variable which you want to annotate in your phenotype data. In a summarized experiment, it's called the column data slot. We are looking into this into a second. So you want to have one variable where you say, what are my cases and what are my controls? Yeah, so where you define the sample groups that you actually want to contrast for differential expression or for enrichment analysis. So for the leukemia data set, the group variable accordingly indicates whether this fusion gene is present, yes, or no. Okay, 1 or 0, binary indication.
So someone wants to know if they can do the normalization using the AFI package
Yes.
for microarray and then input into the image browser.
Yes, absolutely. So the question was, can you normalize your data with other packages beside what is offered here in the enrichment browser? So can you use normalization methods? And there are at least as many normalization methods as there are enrichment methods. And you can apply your favorite normalization method you're very convinced of, and put this into your summarized experiment and accordingly provide it. I just want to say this is a lot easier possible for microarray data than for RNA-seq data when you are using like EdgeR and DE-seq. EdgeR and DE-seq, the question was on microarray data, that's why I said yes. For RNA-seq data, I would say no. When you apply EdgeR or DE-seq, as they rather have their own normalization incorporated in the differential expression analysis. So they really request the raw unnormalized read counts, okay? Good. So if we do that, if we say, okay, we're using here a shortcut, we say, okay, we have this variable which is called group, which we want to define for our expression data set here, and we say, okay, there is a variable which indicates whether this gene is present or not. And according to this variable, we say, okay, this is either a case or a control. And this splits my patient into these two groups. I have 42 patients which are in this respect the normals, and we have about the same number of patients which are my cases here. And these two groups get contrasted. For the airway data set, if we are doing the same, it's rather the question, have these cell lines been treated? Yes or no? Have they been treated with this drug, 1, or have they not been treated with this drug, 0? So I do this in exactly the same way. I say, okay, the group variable for this data set is 1 if it's treated, or 0 if it's not treated with this drug. And this results in these two sample groups for samples each, cases and controls. So there's a lot of experimental design in R. The enrichment browser features very simple experimental designs for two groups. Some extensions are there for paired samples or sample blocks in general, if you have something like badges. And you can also define a block variable for the airway data set. We do this here for demonstration, just like the different cell lines that we have there. There are like 4 different cell lines with 2 samples each. Okay, and then all what we do once we have this group variable annotated, we carry out the differential expression analysis. So we override this expression data set by the expression data set for which we have carried out differential expression. And then we can look up the row data slot. So the row data is one of these 3 matrices of the summarized experiment. And the row data is the one where we have more information on the genes. So we had the expression matrix, we had the matrix which stores clinical information for the samples, and we had this row data matrix which stores information on my genes that I'm investigating. And what do the differential expression analysis do? Well, for all genes that I'm analyzing here, I'm having a log2 fold change, and I'm having a p-value which is adjusted for multiple testing. Okay, so I'm seeing here all of these genes have been tested with a lemma t-test, and I also have the lemma statistic annotated here. And I can do some filtering on it, which genes have a adjusted p-value below a certain threshold in order to get significant genes. Or I could do some filtering on which are like upregulated genes, which have a log2 fold change above 1, for example, which I would consider significantly upregulated. For the airway data set, the RNA-seq data set, we can define particular methods, I said. So there's not only lemma available as for microarray data, but also EdgeR or DSeq2. So here for demonstration, we carry out differential expression analysis using EdgeR. So what does it say? Well, we exclude a lot of genes which do not satisfy a minimum expression threshold. It does not make any sense to do differential expression analysis for genes which are just not expressed at all. So we say, okay, we define a minimum of expression threshold there, which is an argument to that function. So it can be modified. So taking default, it would exclude quite some genes, but there are still over 12,900 there. And for them, we also have now again the log2 fold change, the adjusted p-value, and the HR statistic. And as I said, we can do some filtering and ask, for example, the question, how many significantly differentially expressed genes are there according to HR. So I would ask in the row data, the adjusted p-value slot, this slot here, how many do have an adjusted p-value below this threshold 0.05? And it says, okay, out of 12,900 genes tested, we have roughly 1/3 satisfying this condition. Okay, so I have here a little exercise, and I wonder whether we just take some 5 minutes. I have talked a lot now, and I wonder whether you would like to maybe try things a bit out and just use this function. And as I said, Levi and I are here. We would come around a bit and help in these minutes to set this a bit up. I also show this solution then, no matter whether we managed or not to go through this in this 5 minutes, but I think it's a good idea. So why not trying to do this analysis not only with HR, but rather also like with the lemma-boom method and with the DE-seq method, and ask the same question like this here. How many genes are significantly differentially expressed according to these three methods? Yeah, so the exercise would be compare the number of differentially expressed genes, where we say differentially expressed genes are those which have an adjusted p-value below 0.05 for the airway data set, using these three different main methods for differential expression analysis for RNA-seq data. And whenever there are any problems where we can help, just raise your hand. NumDE, and I say, okay, I provide this function a method name, which is either lemma, HR, and DE-seq. I give this function the expression data set, which is here the airway data set, and I say, okay, this is my significance level, alpha, which is here 0.05. And then I say, do a differential expression analysis of the expression data set using the method given here as an argument. And then for the resulting expression data set with differential expression computed, ask how many genes have an adjusted p-value below the threshold indicated as an argument, and then return this number. And then I say, okay, go over this vector of methods and apply this function numDE to this vector, and the expression data set should be the RE set. So if we do that, so here we apply the function for one method on the data set, taking the default 0.05 alpha level, which takes a bit, but in the end, it returns the number of differentially expressed genes. And then I say, okay, do not do it only for one method, but rather do it for all three. So the sapply function is who does not know the sapply function? So it's basic R. Yeah, so it's just like a for loop. Yeah, but a high level construct in R that you would not say for each element, but you rather say, okay, apply as apply and then go over each element in the vector and do something, which is in this context, this function. Okay. So, and here we have the corresponding number of differentially expressed genes. So we say, see Lima Boom and Azure in about the same order, which is not very surprising as they methodological very similar from the same authors. And then we have DEC, which is a little bit more conservative, and the numbers are a little bit less, but in about the same order. Questions to that? Okay, so let me check a little bit on the time, Levi. How much time is left? We have another hour, so we are pretty good. And I think now it's high time to indeed talk about enrichment analysis here, which we're doing right now. So having the data's differential expression computed and ready, we now want to get the gene sets, and we have talked over and over again about gene ontology and KEGG, and there are the corresponding functions in the enrichment browser named get KEGG gene sets or get go gene sets. And I would say, okay, for which organism here in 3-letter code, okay, I want to have it for Homo sapiens, for human, because both data sets, the leukemia data set and the airway data set are investigating human data. I could also do other organisms here. Here I retrieve the KEGG gene sets, which are there 327. And I also say, okay, give me the Go gene sets for Homo sapiens, and please give me these gene sets from the biological process ontology. Remember, there were like these 3 main ontologies: molecular function, biological process, and cellular component. We are here mostly interested in biological process, and we retrieve it from a bioconductor annotation package, the go.db package. There are a lot more Go gene sets as KEGG, so you see there are like around 11,800. That means here, if you do a Go enrichment analysis, there's a lot more computation to be done. To supply other gene sets instead of KEGG or Go, there's also this possibility to provide gene sets in a very basic text file format, which is the GMT file format, which is nothing more than a tab-separated text file where you have in each line a gene set. And the first element would be the gene name. The second element would be a short description, and then you would have all the genes in this gene set. Okay. And the good thing about it, you would, for example, here take this example file available in the enrichment browser package and say, these are my gene sets of interest. I have derived them from wherever, and I parse these gene sets here from GMT. So these are like some 39 gene sets. Here in this case, it's a subset of KEGG gene sets, just for demonstration. How do they look like? We look at the first 2 gene sets. So this is like the name of the gene sets, and here are like the entree NCBI gene IDs, which are in this gene set. So these are basically, this is basically a list of character vectors. An interesting additional collection you want to consider is the MSigDB, beside Go and KEGG, very frequently used, which is a collection of very different gene sets, either like positional where they are in the genome, also like curated from the literature, motifs where transcription factors are binding, and additional stuff like also cancer-related gene signatures. All you have to do is to register here using this link, and then you can retrieve GMT files for these collections, And after downloading these GMT files, you can read them in using this parse GMT function. So pretty straightforward. Let's do some enrichment analysis. And we start doing the overrepresentation analysis. So this little 2x2 contingency table thing. And we use the function set-based enrichment analysis, which was this umbrella function for a lot of different methods which are there. You see it here in the workflow again. So there is this umbrella function, the facade SBEA, and via this function, a lot of different enrichment methods can be carried out. Okay, so we carry out an overrepresentation analysis on the leukemia data set by calling these functions, saying we want to do an overrepresentation analysis, and we need to have two arguments, two inputs for set-based enrichment analysis. I said the one is your expression data set, so which is here the leukemia data set, and on the other hand, you need the gene sets, which are here these 39 CAG gene sets. And then we are saying something like, okay, this is my significance level, and I do not want to do any permutation of sample labels here. We're doing the very basic standard version of overrepresentation analysis, so just Fisher's exact test. So we're doing that, and then we, on the result, we call the gene set ranking function, and what it gives you back is a table, a capital D data frame, where the gene sets are ranked according to the p-value from Fisher's exact test. And you see, okay, how many genes are in the gene set. So for example, on the top, you see, we're talking here about the leukemia data set, and we are seeing, okay, transcriptional misregulation in cancer is like the most significant one. There are 153 genes in this gene sets, and 17 of them are differentially expressed, okay? So the overlap according to the hypergeometric distribution gives this p-value. So that is like the typical output that you want to get, but the package is called enrichment browser. So there's also a little bit of browsing, so HTML files that you can inspect, which are organized like this. So we have, based on which paradigm you're using for the enrichment, either some illustrative graphics based on sets or some illustrative graphics based on networks. And you also get some information on the differential expression. You have the full flat rankings, of course, but the enriched gene sets, the significant ones, for them we do a little bit more effort in order to really comprehensively visualize and explore them. So why don't we take a look how this would look like. So we have this overrepresentation analysis result, and we call this function called enrichment analysis browsing. So now I need to find my terminal, which is this one, and I need to move this one away. And I say, okay, do that. So it gives me a little bit of message. Okay, I'm creating a gene report. I'm creating some views, and then it should open the browser for me, which is which it does here. Can this be read in the back, or should I increase the font? Can this be read? Yeah. Okay. Better. So how does it look like? Well, we have here the gene sets, which I can rank here interactively. So I want to have them like sorted by p-value. I can go to the corresponding databases. I have here the gene report. where I have like fold changes and adjusted p-value, I could look at the respective genes in the corresponding NCBI gene database. We have, unfortunately, a slow internet connection here, so it takes a little bit longer, but here it is. So there would be some more information. And then for each of these enriched gene sets, we have particular views. So as these are CAG pathways, we have a path view, but in case we have like arbitrary gene sets, you always have a set view. And what you have here on the left-hand side is a interactive volcano plot where you have for each gene in the gene set, the fold change on the x-axis and the minus log 10 p-value on the y-axis. And of course, you're interested in these genes which are like on the top of this volcano. So here we have an upregulated gene, which is apparently also statistically significant. And it's this zinc finger e-box binding thing. And I can click on it and it brings up the corresponding page for this gene here below. So I can like interactively explore which genes I'm interested in. I have a heat map for all genes in my gene set where I have the two groups here indicated, and I see there is another heat map which only shows the significant genes in my gene set here. So you also see here the separation of the samples in cases and controls is a lot clearer for these genes. So far to the set view, let's take a look also on this path view, which is there for CAG genes. So we have on the one hand down here, this laid out pathway map from CAG with the genes which are either up or down regulated. So the upregulated genes in red, the downregulated genes in green. Here directly on the pathway map. From a network-based point of view, I might be only interested in the connected components here of this pathway map, which are shown here on the top. And again, you could like, you might be interested here in this particular gene, then you would click accordingly on it and get the corresponding page in CAG where you get some more information on this gene. Okay. Question? No, that was actually a message, so I better stop my WhatsApp here. I'm sorry? Here. You just execute this command on the enrichment result. So here you have carried out the overrepresentation analysis, right? And you can basically do two things on such a result. You can extract the flat ranking via gs.ranking, or you can call the ea.browse on the result. That should actually work also directly from the console in RStudio. It does not work on the Python notebook here. This is what I know, but I have seen people making it work in RStudio. I always work in a very flat console, so I'm not too familiar with RStudio, but it should work. Okay.
Every time I close my laptop, Zoom disconnects me, so I'm not very much on.
I think there's nothing like but a basic URL browse function below it, which should be able to open the browser also in RStudio. So there's no real magic underlying, but well. Yeah, it's just like browse URL, and browse URL, as far as I know, should be supported in RStudio. Okay, so that's to the browsing. We also want to do such an overrepresentation analysis not only for the leukemia data set, but also for the airway data set. And here we are running into an issue with gene IDs, which you at some point have always when you deal with this kind of data, as there are different resources for gene identifiers. Very prominent ones are like NCBI Entree gene IDs, but you also have like ensemble gene identifier, you have Hugo gene symbols and what have you. So we had the gene sets defined on Entree NCBI gene ID identifiers. However, our airway data set is defined on ensemble gene IDs. That's why I need to map it. I need to map the gene identifiers in my airway data set from ensemble gene IDs to Entree gene IDs in order to be able to subsequently carry out an enrichment analysis. So this is a function from the enrichment browser where I say, okay, I'm working here on Homo sapiens, and by using underneath bioconductor annotation packages again, I say, do the mapping and I get some information from it. So ID mapping is always like associated also with some extent of data loss, and it informs you about it. There were like around 1,000 genes where I had no NCBI Andrea gene, although I had an ensemble ID there. I need to exclude them because there was no information. Yes, please.
Yeah, so what happens with genes? Because when you run this also get matrix selected like this, which I guess is another way to switch.
Get matrix?
I mean, I use also this get matrix with selected IDs, which also changes from, I mean, whatever you want and send them to gene IDs, but you always lose some. But these ones that you lose,
Yeah.
I mean, they are lost because they are not reported or they're not important or not.
All kinds of reasons.
Not coding.
All kinds of reasons. There is unfortunately no trivial, easy solution to a gene identifier mapping. One solution. There is unfortunately no trivial, easy solution to that. The solution would be you sit down for hours, maybe days, weeks, and go one gene by one through it and do the mapping by hand. Typically, you don't want to do that and say, okay, you lose something, but you still have thousands of genes you're analyzing, and there is a lot of signal in these, and yeah, so kind of this kind of logic you're applying there. Yeah.
So the reason why you're losing is just because these are not very well known or they're not coded?
All kind of problems, typically not well annotated in the respective databases. Yeah. So for some, there might be ensemble gene IDs. For some, there might be Entree gene IDs. Really depends on what do these databases do themselves for having these gene identifiers annotated. So all kind of issues. Yeah. And this is like a brute force approach. You say, okay, I'm doing what I can do and what I can do programmatically and systematically. This is what I'm doing. And if you really have an interest to do it yourself, you can do that, but then provide it already by hand. Yeah. So what does it mean? I mean, we are talking about around 10,000 genes and we're losing like 1,000. So we're still talking about 9,000 genes that we're analyzing. But I agree, 1,000 are a lot. Yeah. Yeah. Okay, there are also some genes in ID mapping always for which you have maybe more than one ID that would be able that you can map it to. And again, there is no easy solution to that. There are several options to deal with that. We're taking the approach, we take a single Entree ID for an ensemble gene ID where there would be more than one, okay? So in the end, my airway data set is then mapped on Entree gene IDs, and I can do the overrepresentation analysis as before. saying, okay, here my expression data is the error set, and then taking these gene set, and I can again extract my GS ranking. So if you're a biologist, you might be able to make something out of it. Talking here about CAG, a lot of pathways are having a cancer background. We are actually talking here about asthma. I don't see too much here connected to asthma, which might just be like the reason because there is not a lot of asthma related pathways in CAG. Okay. I think we should go on to gene set enrichment analysis if there are no further questions on the overrepresentation analysis part. Okay. So as I said, gene set enrichment analysis, the great thing is you work on the complete a vector of gene sets. Another thing is you do not assume any particular distribution, but you rather sample your labels of your cases and controls and try to kind of estimate the underlying distribution of enrichment. So you don't have these hypergeometric distribution as before, but you rather take an empirical distribution by permutation. So it's a underlying permutation test statistically. So permutation, it's always the question, how many permutations do you do? If you do a lot, it gets computational intensive, okay? A good number is doing 1,000 permutations, which is a thing you can still do like for microarray data and CAG gene sets in minutes, yeah? So we are running here a gene set enrichment analysis now with 1,000 permutations. And again, we can call the GS ranking function. And we are now getting a enrichment, a p-value, an enrichment score from GSEA, and a normalized enrichment score. So what is it normalized? It's normalized to gene set size. So we're seeing similar pathways. So we were talking about leukemia here. So we're seeing similar pathways about some leukocyte stuff and carcinoma stuff, apoptosis and cancer, which comes up. Okay, one important thing, and that's why I said in the very beginning, we have differences between microarray data and RNA-seq data. And that was this thing with the distributions. In microarray data, we assume a normal distribution, and gene set enrichment analysis was developed for microarray data. So they calculate a local statistic for each gene, which is kind of a t-statistic. And the t-statistic compares the means of two normally distributed variables, okay? If we now apply gene set enrichment analysis to RNA-seq data, we have actually underlying a negative binomial distribution. So I need to modify this t-statistic into a more appropriate statistic which accounts for this distribution.
Okay, what's the distribution?
I'm sorry?
What's the distribution?
For microarrays normal, and for RNA-seq read counts, raw read counts, negative binomial. Yeah. And here for the airway data set, we took HR and a current for differential expression analysis. So the function would check what differential expression analysis has been carried out, and accordingly would repeatedly in each permutation would repeatedly carry out HR on the data set. And this can be then computational intensive because HR and especially DE-seq, they take a lot longer than Lemma. So if you try it out yourself and not just with 39 gene sets, but rather with Go, for example, where you have 11,000 gene sets, this can result in long run times. Okay. So what are alternatives to that? Well, you have several alternatives. One alternative is you do the Boom transformation. The Boom transformation is part of Lemma just due to this reason to unlock methods developed for microarray data where you have the normal distribution for RNA-seq data. So with Boom, we are transforming data which follows a negative binomial distribution to characteristics of a normal distribution. So once I have applied Boom, my data follows the usual normal assumption, and I can apply methods for differential expression analysis and for gene set enrichment analysis as before. This is one alternative. Another alternative is employ an approach which is not that computational intensive. So instead of doing permutations, you can also do some lightweight permutations, which are called rotations. And rotations are used in the roast method, which you can also carry out here in the set-based enrichment analysis function on the raw RNA-seq read counts. And if you try it out yourself, it runs pretty fast, and it's another alternative that you can use. And as I said, and Levi introduced this in the beginning, there are a lot of methods available, and we are also currently analyzing how can they be involved in benchmarking, how they perform. But just for you here as an overview, we took a look at these two methods and a bit on this one, but there are also a number of other methods available here. There is another exercise, yeah.
There was another question. What other packages can do pathway analysis?
I'm sorry?
What other packages can do pathway analysis in addition to
So pathway analysis in the way that we incorporate the interactions, is this what the question was?
Yeah.
Yeah?
I mean, where you can submit a set of genes and get a set of pathways.
Yeah, I think like when it comes to network-based enrichment analysis, this is the next section, so I'm going to answer that during that section. Maybe another little hands-on at that point. I actually would like to say, okay, we have done an overrepresentation analysis for the L data set and for the airway data set. And so far we did an overrepresentation analysis with always these little subset of gene sets. Yeah, these 39. In order to give you a little bit more realistic feeling, I would say, let's take this large catalog here. Let's take these 11,000 gene sets. So execute this command in order to get gene sets from Go for Homo sapiens from the biological process ontology. Use the airway ESET or the ALL data set and carry out an overrepresentation analysis. So the SBEA function method ORA with Go gene sets. And then ask on the resulting gene set ranking, how many are significant? Let's say how many have a p-value below 0.05. Is the exercise clear? Or are there questions about the exercise?
So there seems to be an issue with some RStudio installations, including my own, that I've found a workaround for.
Okay, what is that?
Are there any people having issues with opening the browser in RStudio? Yeah. I'll show you. RStudio open there right now?
No.
It would be easier for me to share my screen.
Are the p-values adjusted in the results?
Very nice question. You can give that as an argument whether and how you would like it to be adjusted. That's a great question. I'm showing these here. So there is an argument which says, okay, what kind of p-value adjustment do you want to do? And the default is do not do any p-value adjustment. So why is that? Well, in gene set enrichment analysis is a lot about ranking. Yeah, you want to see, so you have these two questions. Are they statistically significant and how do they rank to each other? So sometimes they might not even be statistically significant, but you still would like to rank it in order to compare between them, right? And this is what we are currently also like analyzing, how much does multiple testing correction flattens out the signal. I like when I did it empirically myself, and we also have some results on that, found that many multiple testing approaches flatten out completely the signal on my ranking, and I do not have this ranking ability anymore in my rankings. That's why I like to look onto the ranking without adjustment, but I then do not report, okay, I have so many significant or stuff, because then I would need to do some adjustment. Yeah. But here, just to complete on that, you have all the methods available for the p-adjust function in the stats package. So you have Benjamin Hochberg, you have Bonferroni, and what have you.
I'll just copy in my text here, my workaround for people having RStudio issues.
Great,
we'll
that you fixed it
this.
that fast.
Yeah.
That's amazing.
It was, so the command is config.ebrowser.
Wow, you even found this function. This is really hidden in my documentation.
I had to do some debugging. So if you do this, it will give you a directory. You can just copy and paste that directory into your browser, and there's a directory called reports. Go into there. There's a file called ora.html. Click on that, and then you will get everything if it's not popping up automatically.
Thanks, Levi. That's great. Perfect. Cool.
We'll have to figure out what the issue is.
Yeah.
At least it's reproducible. I've got it on my laptop.
Okay. Which is interesting, as it just uses like the regular browse URL in R. So it should actually work with RStudio, but I follow up on that.
Oddly, it works with something like Google.com, but it's not working with the local file.
Interesting. Interesting. Are these all Macs? Ah, okay. So the problem might be that we have this space here. So it might be the case that it does not find it because of this space here, which is only on Mac there. I take a look into that. Thanks for bringing that up.
Yeah, that directory.
Yeah. And there should be a subdirectory reports. And in this report subdirectory, there should be an ORA HTML that you can just click on and find it there. Okay, we were at this little exercise of carrying out and go over representation analysis. So hands up, who would like to continue trying this exercise, or who wants me to continue on the network-based enrichment analysis right now? So hands up for I want to make the exercise. Hands up for network-based enrichment analysis. Okay, we got a majority, great. So you have this exercise for you at home or after the workshop as well. So let's take a look at network-based enrichment analysis. We have 10 more minutes to go, so I make this a bit shorter, but I make sure that we have the main concepts here. So in gene sets, let's take another look at this little pathway map. When we do in gene set enrichment analysis, we basically say we do not take into account all these little interactions which we have annotated and actually in the pathway maps. I just say, I want to see, is there some differential expression? Yes or no. And this often gives me a lot of good conclusions and informations, but I might do this a little bit more sophisticated. I might say, I know that this gene is a transcription factor which activates the expression of this gene. So why don't I score these interactions which I have here? Why don't I ask for something like consistency? So I could ask, for example, if I see this transcription factor upregulated in my expression data, and it correlates with an upregulated target gene here, then I would of course score this positively, because this is a consistent expression change of two genes which rather follows this edge. And I of course can do this vice versa for inhibitors. I could also ask on the main topology, I could ask, okay, I have here a significant upstream change. So I might see this receptor to change significantly, and I might ask how many genes downstream of this receptor are actually changing significantly as well. So this would be another test statistic that I would then do. I would ask how many downstream genes are actually changing. So I take into account these interactions and also the topology of the main pathway. And these are like the main principles that are involved here. So let us look at some example methods. So this is what I say in network-based enrichment analysis. We want to know whether there, for example, are transcription factor and target genes expressed in accordance to their connecting regulations, activation, and inhibition. So let's get a little network. And as I said, this was this file here. A network can be easily compiled by using all these interactions from the CAG pathways. I use the compile.grn function. Which is here. So I have here a little preparation of a selection of CAG pathways, of human CAG pathways, and I say, out of these pathways, please compile a gene regulatory network. And how does it look like? It's a very simple matrix format, which basically says, okay, this is my regulator here in the from column, which are again NCBI entree gene IDs. And in the to column, I have the target genes of these transcription factors. And the interaction type is either activating plus or inhibiting minus. Okay, so we see here, this transcription factor is apparently activating the expression of this target gene. So two methods I want to show here. The one is like signaling pathway impact analysis, which is like specifically applicable to CAG pathways. So it makes really it really takes the information available in the KEGG pathways and do two things. On the one hand, it does a classical overrepresentation analysis on the whole pathway, and on the other hand, it really tests like these downstream effects. It tests, is there an upstream effect which I see in all the genes downstream of the topology of the pathway? And what do I give? Now the NBEA function, it's very similar to the SBEA function. I say, okay, which method do I want to carry out? Which expression data set I'm looking at, which is here the leukemia data set. I again look at these KEGG gene sets, and as an additional input here is the network. And then I run through the SPIA analysis. You see here the pathways processed. And again, I get an enrichment result where I again can call the gs.ranking function and the ea.browse function. So what comes out, again, when I apply a signaling pathway impact analysis, is this gene set table here. And I see not only according to overrepresentation analysis, but also like to signaling pathway impact analysis, I see this pathway as before again, which has now an activation score, the number of differentially expressed genes, the complete size of the gene set, and here the p-value from the signaling pathway impact analysis. So the gene graph enrichment analysis method generalizes this concept from KEGG pathways to like all kinds of networks. So you are not really like only restricted to KEGG pathways, but you can rather define networks in whatever way or where you got them from, and you give them here as the argument, and you will rather evaluate the consistency of each interaction. And this is basically the same. And I want to say some more on the last 5 minutes on genomic region analysis. So I rather skip over this part. Okay, so genomic region enrichment is something rather new. So gene set enrichment analysis is very well established on this differential expression analysis. And the question was, when we have a lot of genomic regions coming out as from particular assays such as ChIP-seq. So we, for example, carry out a ChIP-seq analysis for a particular transcription factor, and we got some regions which are this transcription factor bound to. And then we have these regions across the genome, and we actually would like to know something about them. So again, for example, we would like to know, are there particular features enriched in this genomic regions? So for example, is the GC content enriched there? Do I have promoters enriched there, which would make a lot of sense, right? So it's a transcription factor. So we would like to know, is it enriched in promoters? Is it enriched in genes? So I have basically two region sets. One is my experimental region set, which corresponds to this experimentally derived gene list as before. And on the other hand, I have a genomic region sets which represent some function. And I again do like an overlap and assess this for enrichment, which I could do again with an ORA, for example. And as this picture shows here, it is from ENCODE. There are all kind of different genomic assays which produce such genomic regions experimentally that you would like then to analyze for functional enrichment. And in this context, I found the regional package, a very nice package in Bioconductor, which does a permutation test. So given your experimental regions and your functional region, It says we observe a particular overlap, but how likely is this overlap as compared to random genomic regions which follow some distributional characteristics as my experimentally derived genes. So let's say we have copy number variations detected. Copy number variations detected across the genome. This is my experimental derived genomic region set. And I do an overlap with genes. I want to basically know, do the CNVs overlap more often than expected by chance with genes? And what Regioner does, it repeatedly samples random regions from the genome which follow these distribution of my CNVs. So it would take as many random regions from these chromosomes where these CNVs locate and would repeatedly calculate this overlap. And then it would ask, how often do I observe an overlap as large as the one that I observed, giving me back a p-value for either enrichment or not. So let's look at an example. We are looking at the overlap between CPG islands. This is my experimentally derived one. And we're looking in the overlap with gene promoters. And we know, just like beforehand, that promoter regions are known to be GC content rich. So we expect to see an overrepresentation. So we're using here two genomic ranges sets. The first are here the CPG islands. We take them and we see, okay, this is a genomic ranges data structure. So from chromosome 1 to chromosome Y, these are the corresponding genomic coordinates. And we have the length of the CPG island. We have the counts, how many CPG islands are in such regions, and what's the GC content. So how many bases are either G or C, and what's accordingly like the percentage, how many bases of the total number in here are GC. So this is my one region set, and my other region set are the promoters. And these are the promoters of the HG19 human genome assembly. So I download this as well here from UCSC. And I again have a genomic region set here from chromosome 1 to chromosome Y, which are like at this genomic coordinates. Here, just like for demonstration, we restrict analysis only on chromosome 21 and chromosome 22, which are small chromosomes. So it's this permutation strategy is again computational intensive, but you can also carry out this example if you have a bit more time than on yourself over the whole genome. And what we would do is we would do here this kind of test. So we say, we have on the one hand our CPG islands, which are our experimental regions. On the other hand, we would have our promoters. And I say, okay, please do it on genome HG19 and do 100 permutations. So 100 times draw random genomic regions which are following the distributions of my CPG islands and recalculate the overlap with the promoters. So, and I say, okay, please use the chromosomal distribution of my CPG island. So do not completely sample that randomly, but rather keep the genomic distribution of these regions. And also, if I have several overlaps, just count overlap once. What comes out? It says, if I do evaluate the original region set, so if I have not a random region set, but rather the original CPG island, overlapping with the promoters, I see 720 CPG islands overlapping with promoters. If I look at the permuted data, if I look at random genomic regions, I see this is my distribution if I have random genomic regions. Yeah, so these are over 100 permutations, and the maximum overlap that would have been observed for random genomic regions following the distribution of our CPG islands, it would be 150 overlaps. So by far not the number which I observed for the original region set. And that's why I can conclude, I indeed, as compared to random regions, I see an enrichment of CPG island in the promoters, and with the corresponding permutation p-value, we ask how often do we observe an overlap as large or larger? So not at all. Accordingly, the p-value is as low as we have here. Yes, that's a very great question, which goes into this little side note here. For the function which is evaluated per default in the overlap perm test, it is as easy as one base pair overlap. And I think your question indicates this already. This is a very optimistic definition of overlap. So the cool thing of the regional package is a very flexible package, so you can define a function how you would like to have the overlap defined. So you could also like incorporate your own user-defined functions, and I did that for something like the total genomic size that is actually overlap, which gives you a little bit of a more realistic measure for that. Great question. So this is a permutation test, and my last comment here for this workshop is there is another package which is very nice. It's unfortunately only available for human. The cool thing for regioner is it's available for all organisms where you have these genomes, genome assemblies in bioconductor, and that is a number of model organisms. But there is a package which is there for human and defines functional regions for human, and then does the classical ORA, hypergeometric distribution test between these functional region sets and the experimental ones. And if you are only working on human, you might find this package very useful as well. All right, then I would say thank you for the attention, and if there are any further questions, just let me know.